Hello dear friends, welcome back to another video tutorial from Advanced Biology. Today we will discuss on DNA replication in prokaryotes. We know genetic information must be accurately copied every time a cell divides, and DNA replication is one of the process of copying genetic information. A common type of replication that takes place in prokaryotic circular DNA, such as that found in E. coli and other bacteria, is called as theta replication because it generates a structure that resembles the Greek letter theta. British physician and molecular biologist John Cairns provided the first visible evidence of theta replication in 1963 by growing bacteria in the presence of radioactive nucleotides. During prokaryotic cell division, at the end of DNA replication, from the parent DNA, two different daughter DNAs will be produced. So there are some noticeable features of DNA replication. The process is semi-conservative, bidirectional, and semi-discontinuous. The replication or polymerization process occurs in 5' to 3' direction. There is proofreading activity of the DNA polymerase enzyme. We will know here that there are different proteins, many types of enzymes including various DNA polymerases are involved in DNA replication. The DNA synthesis process requires deoxynucleoside triphosphates and a primer template junction. Well, semi-conservative means each new DNA molecule having a parental strand or old strand and a new strand. So half of the genetic material or information which is retained in a newly synthesized DNA molecule, that is known as semi-conservative DNA replication. The two strands of parent DNA molecule acts as a template for the synthesis of new DNA molecule. What do you mean by template? Template means it is like a mold. It allows the molecule to be synthesized in a specific order. That's why it is known as template. So these parent DNA strands act as template for the synthesis of new DNA molecules. That's why it is known as semi-conservative, and it was proved by the experiment done by the scientists Methylation and Stoll by using two isotopes of nitrogen, N15 and N14. They proved that this DNA replication is semi-conservative in nature. Now this replication, it proceeds bidirectionally in 5' to 3' direction towards the fork and away from the fork. The semi-discontinuous, which means the synthesis of new DNA molecule occurs in continuous manner in one strand, while in discontinuous manner in the form of fragments in the other strand. That's why it is known as semi-discontinuous. Considerable research has focused on the origin of replication in E. coli. The origin is named as ORICI for origin of chromosomal replication. It has been estimated that the E. coli origin of replication spans approximately 245 base pair of DNA. Three types of DNA sequences are found within the ORICI: an AT-rich region, DNA box sequence, and Gatc methylation site. DNA replication begins with the binding of DNA A proteins to the sequences within the origin of replication known as DNA A boxes. The DNA A boxes serve as recognition sites for the binding of DNA A proteins. When DNA A proteins are in the ATP bound form, they bind to the 5 DNA A boxes in ORICI to initiate DNA replication. DNA A proteins also bind to each other to form a complex. Other DNA binding proteins such as HU and IHF also causes the DNA to bend around a complex. DNA A proteins, which results in the separation of the two strands at the AT-rich region, and the process is also helped by the HU protein. Because only two hydrogen bonds formed between AT base pair, whereas 3 hydrogen bonds occur between G and C. The DNA strands are more easily separated at the help of at the AT-rich region Following separation of the AT-rich region, the DNA A proteins with the aids of DNA C proteins recruit the enzyme DNA helicase to this site. When a DNA helicase encounters a double-stranded region, it breaks the hydrogen bonds between the two strands, thereby generating two single strands. Melting of the helix initiates a series of events that construct a nascent replication fork at either end of the open region. The first step is the attachment of pre-priming complex at each of the two positions. Each pre-priming complex initially comprises 12 proteins, 6 copies of DNA B protein and 6 copies of DNA C protein. But DNA C has transitory role because it is very much transient and it is released from the complex soon after it is formed. Its function probably being simply to aid the attachment of DNA B. The Gatc methylation sites within the Orishi are involved in regulating the process of DNA replication. These sites are methylated by an enzyme known as DNA adenine methyl transferase but DAM. Prior to DNA replication, the Gatc sites are methylated in both strands. This full methylation facilitates the initiation of DNA replication at the origin. Following DNA replication, the newly made strands are not methylated because adenine rather than methyladenine is incorporated into the daughter strands. The initiation of DNA replication at the origin does not readily occur until after it has become fully methylated because it takes several minutes for DAM to methylate the Gatc sites within this region. DNA replication does not occur again too quickly. We find here the AT-rich region is composed of 3 similar sequences that are 13 base pair long and highlighted in blue. The 5 DNA A boxes are highlighted in orange. The Gatc methylation sites are underlined here. 2 DNA helicases begin strand separation within the Orishi region and continue to separate the DNA strands beyond the origin. These proteins use the energy from ATP hydrolysis to catalyze the separation of the double-stranded parental DNA. In E. coli, DNA helicases bind to the single-stranded DNA and travel along the DNA in 5' to 3' direction to keep the replication fork moving. The DNA B or helicase is required for the unwinding of the double-stranded parental DNA molecule. It causes processive unwinding. Processive means moving forward or progressive. At this point, single-stranded binding protein or SSB is required to hold the two strands, preventing the reunioning of separated DNA strands. Next one is the topoisomerase group of enzymes, which include topoisomerase 1 and topoisomerase 2. These enzymes are required to relieve the torsional strain created by the helicase-induced unwinding. The supercoils, which are produced by the helicase unwinding, show the topoisomerase has both the nuclease as well as ligase activity. Topoisomerase 1 nicks the single strand. Here we can find the difference between two types of topoisomerase, topoisomerase 1 and topoisomerase 2. Topoisomerase 1 nicks the single strand, relieves the supercoil and again ligate it, while topoisomerase 2 nicks both the strands and also relieves the supercoil and then again ligate it. After this, the primase is required for the synthesis of RNA primer because RNA polymerase cannot synthesize at its own. So what is RNA primer? It is a short stretch of ribonucleotides which is complementary to the template DNA strand. The initial segment of nucleotide, it is not synthesized by the DNA polymerase, it is synthesized by the RNA polymerase. So the primase is the DNA dependent RNA polymerase and is required for the synthesis of RNA primer. The chemistry of DNA synthesis requires that the new chain grows by extending the 30 end of the primer, 3' end of the primer. The phosphodiester bond is formed is an SN2 reaction in which the hydroxyl group at the 3' end of the primer strand attacks a phosphoryl group on the incoming nucleoside triphosphate. The leaving group for the reaction is a pyrophosphate, which is composed of the beta phosphate and gamma phosphate of the nucleotide substrate. There are basically 5 types of DNA polymerase in prokaryotes, which are DNA polymerase 1, 2, 3, 4 and 5. DNA polymerase 1, encoded by polymerase A, is involved in repair of damaged DNA and in a subsidiary role in semi-conservative replication. DNA polymerase 2 is required to restart a replication fork when its progress is blocked by damage in DNA. DNA polymerase 3 is the major enzyme involved in replication process, which is also known as replicase. It is the product of DNA C protein. DNA polymerase 4 and 5 are involved in allowing replication to bypass certain types of damage, and they are called error-prone polymerase. Only one DNA polymerase is the replication enzyme, that is DNA polymerase 3. The others participate in repairing damaged DNA, restarting stalled replication fork, or bypassing damaged DNA. In 1968, a paper in PNAS, Rezi and Tyushenko-Okazaki and their colleagues proposed that the lagging strand is replicated discontinuously in the form of small fragments. Later, these small fragments were named as Okazaki fragments. Now, when DNA replication elongation occurs, one of the strand is synthesized in continuously. That strand is known as leading strand, and the other strand is synthesized in a discontinuous manner. So there are many small fragments are produced. This strand is known as lagging strand, and the smaller fragments were named as Okazaki fragment. So there is DNA polymerase, which shows the 5' to 3' polymerization activity. So each successive nucleotides or newly incoming nucleotides are added to the 3' OH of the previously added nucleotide. So polymerization activity or DNA synthesis occurs in 5' to 3' direction. The DNA synthesis occurs in two strands. One strand is leading strand and the other one is the lagging strand. We know this. One strand synthesis occurs towards the fork, that is the leading strand, and other one is away from the fork, that is the lagging strand. Then we will see the proofreading activity, which is another function of DNA polymerase 3 in prokaryotes. Whenever there is mismatch base or any incorrect base is added to the growing DNA molecule or newly synthesized DNA molecule, the base is corrected by the DNA polymerase 3 molecule. DNA polymerase slide back to the original position, remove the incorrect base, and then the correct base is added to that particular position. The purposeful process occurs during DNA replication is known as proofreading activity. The proofreading function belongs to DNA polymerase 3 enzyme. DNA polymerase 3 is the major enzyme involved in the replication process with highest polymerization rate. Polymerization rate means number of nucleotides added per second. It is near about 250 to 1000 nucleotides are added per second by DNA polymerase 3, and it has maximum processivity. Processivity means number of nucleotides added before the DNA polymerase 3 enzyme dissociates from the template DNA complex. So it has more than 5 lakhs processivity, which means that more than 5 lakh nucleotides are added before the DNA polymerase 3 enzymes dissociation from the template DNA complex. So DNA polymerase 3 is the major enzyme in prokaryotic DNA replication process, but DNA polymerase 1 has a unique characteristic feature. That's why it is very important. The feature is DNA polymerase 1 has 5' to 3' exonuclease activity. It is required for the removal of Rna Primer, and this activity is only found in DNA polymerase 1. It also has a 5' to 3' polymerase activity, which is required for the synthesis or adding of the nucleotides. Then it also has 3' to 5' exonuclease activity, which is required for the proofreading function. So DNA polymerase 1 has all the three activities: 5' to 3' polymerase activity, 5' to 3' exonuclease activity, and 3' to 5' exonuclease activity. But the DNA polymerase 3 has 5' to 3' polymerase activity and 3' to 5' exonuclease activity. It has no 5' to 3' polymerase activity. So DNA polymerase 2, which we now know mainly involved in repair of damaged DNA rather than genome replication. DNA polymerase 1 and 2 are single polypeptides, but DNA polymerase 3, befitting its role as the main replicating enzyme, is a multi-subunit protein with a molecular mass of approximately 900 kilodalton. Then, after primage, we require DNA polymerase 3 enzyme. It is needed for the polymerization and proofreading activity, means addition of nucleotides, and to repair there is any mismatch. After DNA polymerase 3 activity, DNA polymerase 1 is required, and it is required to remove the RNA primer due to its 5' to 3' exonuclease activity. After DNA polymerase 1, DNA ligase enzyme is needed. In E. coli, DNA ligase requires NAD+ to carry out this reaction, whereas the DNA ligase found in archaea and eukaryotes require ATP. DNA ligase is a nick sealing enzyme, so these are the various enzymes required for replication process. DNA polymerase slides along the template strand as it synthesizes a new strand by connecting the deoxyribonucleoside triphosphates in the 5' to 3' direction. The catalytic subunit of DNA polymerase resembles a hand that is wrapped around the template strand. Thus, the movement of DNA polymerase along the template strand is similar to a hand that is sliding along a rope. Now, we understand that how the leading and lagging strands are prepared or made. The figure here shows how new strands are constructed from a single origin of replication. To the left of the origin, the top strand is made continuously in the right side image, whereas the right of the origin, it is made in Okazaki fragment. By comparison, the synthesis of the bottom strand in this right side image is just opposite. To the left, to the left of the origin, this strand is made in Okazaki fragment. To the right of the origin, the synthesis is continuous. DNA strands, what happens, separate at origin, creating two replication forks. Primers are needed then to initiate DNA synthesis. The synthesis of the leading strand occurs in the same direction as the movement of the replication fork. The first Okazaki fragment of the lagging strand is made in the opposite direction. So the leading strand elongates and a second Okazaki fragment is made. The leading strand continues to elongate. A third Okazaki fragment is made, and the first and second are connected to each other. Bruce Alvarez first proposed that DNA replication occurred within a large replisome complex with coupled polymerases catalyzing leading and lagging strand synthesis. At the same time, anchored to the replication fork helicase, elements of this model, which is also known as trombone model. Trombone is a musical instrument. Here is the image of the trombone. Have been confirmed in a number of systems. Leading and lagging strand polymerases are coupled but have asymmetric properties. DNA loops predicted to be formed on the lagging strand can be observed by electron microscopy, and the lagging strand polymerase recycles to continue synthesis without dissociation from the fork. The first step here, the DNA helicase denatures the parental duplex DNA and single-stranded binding protein SSB protein coats the leading strand and lagging strand templates. The leading strand DNA polymerase 3 sliding clamp complex synthesizes the leading strand continuously. The lagging strand polymerase III sliding clamp complex synthesizes an Okazaki fragment. The primase binds the lagging strand template and synthesizes a new RNA primer. SSB or single-stranded binding protein is released ahead of leading strand and lagging strand synthesis and ahead of RNA primer synthesis. Lagging strand DNA polymerase C comprises synthesis of an Okazaki fragment and is released by the sliding clamp. A DNA polymerase 1 replaces the DNA polymerase 3 to begin removal of the RNA primer and replacement of RNA nucleotides by DNA nucleotides. The fourth point, DNA ligase joins the Okazaki fragment. The clamp loader places a new sliding clamp near the 3' end of the RNA primer on the newly primed lagging strand. Lagging strand DNA polymerase 3 binds the sliding clamp and initiates the synthesis of a new Okazaki fragment. This is all about the trombone model for DNA synthesis, the elongation process of DNA replication. DNA helicase and primase are physically bound to each other to form a complex known as primosome. This complex leads the way at the replication fork. The primosome tracks along the DNA, separating the parental strands, and is synthesizing RNA primer at regular intervals along the lagging strand. Being associated together within a complex allow the actions of DNA helicase and primase to be better coordinated. The primosome is physically associated with DNA polymerase holoenzyme to form replisome. Two DNA polymerase III proteins act in concert to replicate and the leading and lagging strands. The term dimeric DNA polymerase is used to describe two DNA polymerase holoenzymes that move as a unit during DNA replication. For this to occur, the leading strand is looped out with the respect the DNA polymerase and that synthesizes the lagging strand. This loop-like structure allows the lagging strand polymerase to make DNA in 5' to 3' direction, yet move as a unit with the leading strand polymerase. Interestingly, when the lagging strand polymerase reaches the end of the Okazaki fragment, it must be released from the template DNA and hop to the RNA primer that is closest to the fork. The clamp loader complex, which is part of DNA polymerase holoenzyme, then reloads the holoenzyme at the site where the next RNA primer has been made. Similarly, after primase synthesizes an RNA primer in 5' to 3' direction, it must hop over the primer and synthesizes the next primer. Replication is terminated when the replication forks meet at the termination sequence. On the opposite side of the E. coli chromosome from Orishi is a pair of termination sequences known as the TER sequences. A protein known as the termination utilizing substance or TAS binds to the TER sequence and then stops the movement of the replication forks. One of the TER sequence designated as T1 prevents the advancement of the fork that is moving left to right but allow the movement of the other fork. Alternatively, T2 prevents the advancement of the fork that is moving right to left but allow the advancement of the other fork. In any given cell, only one TER sequence is required to stop the advancement of one replication fork, and then the other fork ends by its synthesis of DNA when it reaches the halted fork. In other words, DNA replication ends when oppositely advancing forks meet, usually at T1 or T2. Finally, DNA ligase covalently links the two daughter strands, creating two circular double-stranded molecules. After DNA replication is completed, one problem may exist. DNA replication often results into intertwined DNA molecule known as catenane. Fortunately, the catenanes are only transient structure in DNA replication. In E. coli, Topoisomerase 2 introduces a temporary break in the DNA strands and then rejoin them after the strands have become unlocked. This allows the catenanes to be separated into individual circular molecules. That's it, and I believe that the video will help you all. If you like this video, please hit the like button and share this video with your friends. If you have any doubt, you can comment on this video. Subscribe to this channel to get more and more video like this in future. I will see you in the next video. Thank you. Bye.